Alright, Thursday night, folks. This will serve as your lecture for Friday. Be the last in this kind of series of lectures about systematics, phylogeny. Didn't really think it was appropriate to move on without talking about, what that would look like to build a phylogeny using the information in a DNA molecule. Okay, so it's really about phylogenetics. And these are the main steps that you'd have to follow in order to do this. And so the first thing you have to do is get some tissue samples. Salamanders, it's easy. You just clip a little of their tail off. And we know that they grow that back without really much effect on the organism. Fish, you can take a little clip of the fin. You can take hair is a little bit more challenging for mammals. But there's all different ways that you can get that tissue. So you get the tissue, you have to isolate the DNA out of the tissue, okay? Because remember that tissue is made up of a whole bunch of different things, right? Including some cells, but you have to get into the cell and get that DNA out and get a nice pure product. Then we take the long chromosome and we pick one region on the chromosome, usually a gene, maybe a partial gene, and we will amplify that. So we're going to take that one section and make many copies of that section. We're going to really determine the sequence of nucleotide bases within the gene, and then we're going to bring that sequence. We don't do the sequencing. We'll probably send that off to a lab. They would send back like what amounts to a text file that has all those A's, T's, C's, and G's for all of our different samples, and then we align those so that base pair 1 is the same for all of our samples, and then we use the information. This is really similar to that handout that I gave to you on Wednesday, looking at the 4 lizard species and all the information encoded in there. Okay, then we use a computer to generate those trees. Okay, isolating the DNA. You use a detergent, strong detergent, to rupture the membranes. Membrane is just a lipid, so we're going to use basically the same kind of thing you would use to wash grease off of your dishes, would be to break that lipid membrane down. Then remember that DNA is wound up around those protein histone cores, so we use a type of enzyme to break down the protein. The enzyme attacks proteins. It's not going to affect the DNA because DNA ain't a protein, right? Then we take that DNA that is in solution and we put some very cold alcohol and it will precipitate that DNA out, and then we use that purified DNA and we put it into PCR, which you guys, I know everyone learned a little bit about this in Bio 120, polymerase chain reaction. This is basically a Xerox copy for a specific part of a chromosome. So if we want to amplify or take a small amount of a given gene and amplify and make it bigger, make many copies of it, PCR is the way to go. And in fact, these days you may hear about COVID testing and whether it is a PCR test. So what they're doing is they're looking for specific genes in the RNA that is contained in the virus. So if that specific sequence is there, then it tells you that the virus is there. So you take the DNA from the last step, the isolated DNA. We have to have these things called primers, and then we have this. This is another molecule, Taq polymerase. Interesting long story there. This is a polymerase, a DNA polymerase enzyme from a type of bacteria that lives in hot springs. So what does polymerase do? It takes and makes polymers. It uses the monomer nucleotides and adds them together into a long chain, a polymer, which is a nucleic acid, which is a copy of the gene that we're interested in. So here's the DNA that we put in at the beginning, came out of our DNA isolation. Put it in, you heat it up. When you heat it up, it doesn't break down the polymerase enzyme because it is accustomed to working in hot springs. It does not break down the DNA, but it breaks those hydrogen bonds between the bases and opens up that double helix into 2 individual strands. Primers get put on. So what the primer is, basically it's a starting point for the polymerase. You need a little primer there, then that enzyme polymerase hops on to the primer, and it makes a complementary copy of that DNA. Right, here you go. It's making a copy. 3' to 5', 3' to 5'. Here we go. Now we have 2 copies. Right? Blue is the new version, and red is the conserved old copy. And guess what we do with these 2? Put them back in at the beginning. We heat them up, separate them, attach primers, extend from that primer with a polymerase. Then we have 4, and then we have 16, and then we have, right? So exponential. A few cycles of this, and you have thousands of copies of the gene that you want. Okay. Then we take that product of the PCR and we send it off. Right? Here's PCR is another way of looking at that. Right? So you basically put in our original, separate them, we make a copy of each, and millions of copies can be put out in an hour, not a year. So the PCR product is then sent off to a lab, because what we don't know is the sequence in all of these copies. This is all copies of the same thing. Send it off. There is a machine, a sequencer, a Sanger sequencer, that fires some type of energy at the dyed DNA, and based on the wavelength that comes back, it tells the machine for a given base pair whether it is a G, an A, a C, or a T. Okay, so this will give us the actual sequence of nucleotide bases that occur in that given gene. Okay, and that is the information. We want to see how mutations have accumulated within given groups, and how those mutations differ from mutations in other groups. Right? And that's going to allow us to see which group is more similar to each other, right? And which group is more similar to the outgroup. Whichever group is more similar to the outgroup, we're going to assume is the oldest group, right? Because we know that the outgroup is most distantly related. And that allows us to make those trees. In the Salamander paper you guys read, the Site B tree was made exactly like this. The genomic data is a little bit different, a little bit the same. Basically gives you many, many more pieces of information. Same idea though. Okay. And again, why do we make those trees? Why do we make those phylogenies? It is about describing the evolutionary history of a group, and therefore using that to inform how we name and classify those groups. Okay. We said already, monophyletic is cool. We can use monophyletic groups. We can name them as really kingdom, phylum, usually probably something like a genus. We can use it to name species. Just like with the salamander paper, all the yellow-spotted whirly eyes formed a good clade that had one single common ancestor that was different from all the other whirly salamander, good monophyletic group. These other two, you can't leave people out, and you can't just cherry pick the best from two different groups. Right? This is a group, that's a group. You can't put B and C together. We've talked about all this. So I want to to talk about a couple more examples of how this has been specifically used in vertebrate zoology to inform taxonomy and classification. So this story goes, this guy, I'll say maybe it was Bill Peterman. He was a grad student of Ray Semlich's, who was sort of like an amphibian god. And I think it was Bill. So he goes, is traveling through Georgia to go, I think, Savannah Riverside or something up in South Carolina. And he stopped overnight and was camped out sort of on the edge of the Piedmont, edge of the Blue Ridge, over in Habersham County, I think, where far eastern Georgia. And he goes out at night and does some collecting, collects these little salamanders, sends them off to my friend Carlos. Carlos Camp is the guy that was on the Whirly Eye paper with me. And Carlos doesn't think much about it. He thinks it's probably just like a juvenile of the dwarf salamander. And so he was going to preserve them and put them in the collection at Piedmont. And when he flipped the baggie over, he said, "Wait, what the heck is that?" He noticed this little immature, quote-unquote immature salamander actually had eggs and was a mature, full-grown adult. Okay, this is the size of a full-grown adult of this group. And so, I mean, Carlos is very knowledgeable about salamanders, and so he was just totally floored by this. So they take some of the tissue and they send it off. Well, he isolates the DNA, amplifies a given gene, sends it off for sequencing, gets the sequence back, makes a tree, and including a whole bunch of other species of salamanders that in the same group, that dwarf salamander we talked about, other ones. And this is the tree that they got back. Okay, Quadridigitata, this is the dwarf salamander he thought it was originally. This is the two-lined, cave, long-tailed, three-lined, and then there's these weird neotenic ones from out in the Ozarks. This is Eurycia, okay? This is a genus of salamanders that looks very, very similar to this species. If you had this guy, besides the fact that it's so tiny, it looks just like a two-line salamander. When they looked at this and they got back the tree, it was clear what they were dealing with was something entirely different than any of the Eurycia. So in fact, they considered this to be more different from all the other Eurycia than you would need to really, in order to justify assigning it to another species of Eurycia. So they in fact named it as an entirely new genus of salamanders. The genus is Urspilurpes, and the species name is Brucei, after this guy, Richard Bruce. So Urspilurpes, boom, was born. And again, the point of this, why we're talking about it, besides me trying to indoctrinate you about how cool salamanders are, is the idea that this tree is our hypothesis about relatedness, evolutionary history, and then based on that, you decide how to classify this animal. Okay, really weird animal. They've now been found in South Carolina, but it's really almost endemic to Georgia. Okay, another interesting story happens to come from the herp world. When I learned all of these scientific names, like you guys are, when I learned these are those little blue-tailed skinks, we see them all around. It would have been Eumeces fasciatus, Eumeces laticeps. That genus for that group would be Eumeces. But the weird thing was, and that's always sort of made me wonder, when you go to Japan and you go into the forest there and you catch a little lizard that has a blue tail and it has stripes on it. And when the breeding season comes, they get a red head. It's obviously a skink. It looks just like our skinks. And they were in the same genus, Eumeces, which, Japan used to be relatively closely connected to the North America when continents were in different positions on the Earth. But nonetheless, that's kind of surprising. Made it easier to remember the names of these Japanese groups. So for a long time they remained in the genus Eumeces. However, when they collected a bunch of tail tips from all around the world from blue-tailed skinks, and they isolated the DNA, and they amplified the gene, and they sequenced that gene, and then they probably did this for several genes, and they made trees. Right? This tree shows us a bunch of, these are all lizards. The outgroup here, Tachydramus is another type of lizard. And then this group here really are this kind of skinkid type of animals. And I think, yeah, we're going to focus in on this part, okay? Because here's Eumeces, Schneideri, Schwarzii. These are all the Japanese and Asian species of Eumeces. Here are our species of Eumeces. Fasciatus, this one lives on our campus, Eumeces fasciatus. And what we see, clearly we see in this tree, is that if you continue calling all these skinks a Eumeces genus, it is not a good monophyletic group. Okay? It in fact would be this would be a polyphyletic group. And in cladistics, it can't fly. We cannot continue to call this Eumeces over here and this Eumeces over here. Now, what you could do is say, you could say, basically, well, no, I mean, you'd have to call this, and everybody from here down to here, and including all these, would all have to be Eumeces. Or, what you do, right, alternatively, you say these guys, now this is where we talked about priority on Wednesday. Say, basic tenet of taxonomy is the first group to get that name gets the priority, okay? And the first Eumeces named were the ones in Asia, so then that leads to the situation where you need to rename this monophyletic group here, right? All of these species, what we're calling Eumeces, now become a new genus called Pleistodon. Okay, these guys stay Eumeces. Now, when you catch a skink on campus, it's Pleistodon fasciatus, Pleistodon laticeps, or we talked about this Pleistodon inexpectatus, which is the southeastern five-line skink. So again, another example. Here's Elaphie, same thing, right? Here is the North American. This is corn snakes, box snake, milk rat snake, all in the genus Elaphie until probably 10 years ago. Here is these different species found in Asia of Elaphie. Right? Make a tree, zoom in on this, we see Elaphie up here. So this group here, you could say, okay, that's cool. Monophyletic group, Elaphie. But then, down in this group, right, all of these used to be Elaphie, but they've been now reassigned to a new genus called Pantherophis. It's like the panther snake, Pantherophis, right? Because you can't have Elaphie here, Elaphie down there, and in between pine snakes and ground snakes and milk snakes. I mean, here's the darn king snake. So that makes no sense at all to have Elaphie here and here. So that's a total reassignment. So what that tells you, and this kind of blows my mind, especially with the skinks. What this tells us is that being this exact shape, having a blue tail, having a little striped morphology or phenotype, a color pattern, is a homoplasy. It is evolved independently in Asia and in North America, right? So, i.e., that phenotype popped up here, and it popped up down here, and over here, and certainly the common ancestor wouldn't have looked like that, because common ancestor includes all these other species, right, that don't look anything like that. So that's a homoplasious character state. And that's it. Okay, so another couple of examples, and I may on the exam, I probably am going to give you a phylogeny, and I may or may not point out some of the taxonomic issues based on the taxonomy, or based on the phylogeny. But if you look on here, if you look at this one, if I give you this phylogeny right here, say I don't put this little box on here, and I just ask you, damn it, if I ask you what's wrong with it, okay, then you should be able to, what the hell's wrong with it? There we go. So if I give you this, and I said, according to somebody who follows cladistics, what's wrong with this tree? Well, you should be able to look and see, right, Eumeces here, Eumeces down there, and in fact, here's another genus that's popping up in the middle of the Eumeces, right? This is just a mess. This is a taxonomist nightmare. And so what would the alternative be? Well, you could call this, there's other issues that would have to be addressed here, because even the Asian Eumeces are polyphyletic, because you've got Eumeces here, and then we got this thing called Scincopus, and then Scincus, and then Eumeces over there. It's really just a hot mess. So, be looking for monophyletic groups, be looking for non-monophyletic groups, and thinking about how you could change the naming schematic of a group in order to bring it into a monophyletic condition. Okay? And our most powerful tool for this and is molecular phylogenies based on single genes, multiple genes, but really it's the new gold standard is becoming this, looking at the entire genome of animal and then using that to make these trees. Okay? And that is what's causing most of name changes right now, that we're seeing shifting of classification is because of our different understanding. We assumed that all of these Eumeces from the US and Japan were more closely related to each other than they would be to all these other groups, but that proves to be totally inaccurate, right? And now, 10 years, you may have all of the Asian species renamed, right? Taxonomy is always changing based on new evidence, okay? So that's the lecture for Friday, what is that, August 28th. And then next week we're going to jump in and start to talk about what is a vertebrate, what are those as a monophyletic group, as a big a clade, what are the synapomorphic conditions that unite them as a group, what are the shared derived characters that they have that makes them different from other groups and makes them a good group. Okay, and we're gonna talk about that, and then we're gonna start jumping into some of the nitty gritty of some specific groups of vertebrates. Okay, so we'll see you next week.